The Business of Biotech is produced by Life Science Connect and its community of learning, solving, and sourcing resources for biopharma decision makers. If you're working on biologics process development and manufacturing challenges, you need to swing by bioprocessonline.com. If you're trying to stay ahead of the cell or gene therapy curve, visit cellandgene.com. When it's time to map out your clinical course, let clinicalleader.com help. And if optimizing outsourcing decisions is what you're after, check out outsourcepharma.com. We're Life Science Connect, and we're here to help. Considering that about a month ago, the CDC reported that just 8% of U.S. children and 21% of U.S. adults were up to date with the latest COVID-19 vaccine, which at the time had been available for a few months, it's no surprise I brought the virus home from the J.P. Morgan Healthcare Conference. Between COVID fatigue and vaccine hesitancy, getting into the COVID vaccine game right now might, at face value, seem like a bad business move. That is, if you're coming at the COVID vaccine game from the same perspective Moderna and Pfizer-BioNTech did back in 2020. But maybe if you bring a new vaccine paradigm to the population, you'd find that COVID fatigue and vaccine hesitancy are actually the driving forces behind commercial acceptance of your product. I'm Matt Pillar. This is the Business of Biotech, and I'll never knock the incredible result of the public-private partnership that we saw during Operation Warp Speed. But to cut to the chase, there's room for improvement on many fronts. We all know the first iteration of new technology is never the best iteration of a new technology. I won't put words in his mouth, but Dr. Carsten Rudolph, CEO at Ethris, agrees with at least some of this sentiment. Otherwise, he wouldn't be taking his company down the path of developing intranasally administered and inhaled vaccines and therapeutics for a number of respiratory indications, COVID among them. I caught up with Dr. Rudolph in San Francisco to tape today's episode. Let's give it a listen.
That's where I want to start. I want to start with a little bit of background on where you came from and how you got into this space. So obviously I did a little bit of research. I trolled crept up on your LinkedIn profile a bit. And I learned that you have a pharmaceutical degree and have quickly, on the heels of that, you dove into the mRNA space. I mean, at least it appears that way.
What does mean quickly, So I did my Ph.D. exactly at the Department of Pediatrics, basically in Munich, and at the Ludwig Maximilians University. Mm-hmm. And there, my Ph.D. I was mostly focusing on developing a gene therapy approach for the treatment of cystic fibrosis, because cystic fibrosis, of course, it's in a specialty or university pediatric hospital. It's a quite a prominent disease, Mm-hmm. And so this was always my idea to do research in providing a new potential cure for these patients by delivering genes, of course, to replace the missing function in the body of these patients. But I was mostly working non-viral gene delivery. That means just using plasmids. Mm-hmm. And we all know. And I think you're also a biologist, aren't you?
No. Far from it.
Okay.
Dr. Rudolph, I should have set that record straight from the outset.
But then I'll give you a brief biology tutorial.
Well, listen, you've got to toe the line here, because I am not. Listeners of the podcast know I've said it many times. I dropped out of advanced placement biology my senior year of high school.
Okay. All right.
However, our audience will appreciate a little bit of flavor, So,
but one really problem for using, that's plasmid DNA. Then of course is how to get the plasmid into the nucleus, because the gene, of course, is active in the nucleus. And the lung tissue is a quite post-mitotic tissue, so it's not fastly proliferating, where then the nuclear membrane breaks down, basically. So it's really a limitation. And then we thought, and we knew in those days, basically, that our delivery systems, the nanoparticles that we used, we could pretty well shuttle the messenger RNA into the cytoplasm. So that worked quite well. Mm-hmm. And then we thought, is there a nucleic acid, basically, that you could use for the purpose that is active in the cytoplasm. And of course, that's the messenger RNA, that then directly can be translated at the ribosome to produce the missing protein. Then this was then the idea, well, why shouldn't we use messenger RNA? In those days, no one was really doing a lot of work in this space here. And we started on this and we saw, well, that works really well. They're much, much more efficient than using plasmids here. But we saw at the same time, that just a standard messenger RNA molecule, and we know this all now from the pandemic vaccine development as well, it's quite immunogenic, because we have a lot of sensors in our innate immune system that is made for detecting these foreign RNA molecules, because this could signal just invasion of the virus, like the coronavirus, which is an RNA virus. So we thought, well, it's very efficient, but it's also quite immunogenic, basically. And then we thought, how can we navigate around this? And then we thought about modifying the messenger RNA and introducing modified nucleotides, because we knew from the siRNA field and the oligonucleotide field in those days that if you do so, you can really reduce the immune response and recognition of the RNA molecules. And then we played a little bit around and we found really a scheme where we can change the pattern of the RNA molecules so that it's not recognized anymore that well by their immune system. And then we thought, well, that works quite well. Shouldn't we exploit this potential of such molecules? And then we started to found the company. So that was the idea behind it. So.
Yeah, that's why the question I have for you is you were talking about this research work and we, like who was we at the time, like pre-Ethris?
Pre-Ethris, this was my research team basically at the Department of Pediatrics and my colleagues over there. And I shared the lab space with another professor, and we were running those experiments in these days. And then, I was always, because we all know, that the RNA molecule itself, the naked RNA molecule, is not well taken up by the cells there. It does not penetrate through the cell membrane. And so you always have to package this basically into nanoparticles and then taken up to release the mRNA payload in the cytoplasm. And I was always also intensively collaborating with now also my co-founding partner of the company, Christian Planck, who was running a research group as a professor at the Technical University in Munich. So that's their second large university in Munich. Mm-hmm. And he is a really world-leading expert in designing carrier systems for nucleic acids. And then we thought basically, we can put this together, the expertise, and found the company. And then after we got the first business angel convinced, then we were really brave. So, no, we have to do it. So, I will always remember this moment. It's a funny moment. So my plan was just to do something. I wanted to become a full-time professor, basically, but now I became an entrepreneur. So it's
I want to, you brought it up. So now I want to dig into it. You made that decision. So there are many questions there. What was your biggest reservation? So you thought, I'm destined for professorship. And then you did this work and you realized, well, there are the foundations of a biotech here. What was your biggest reservation about making that leap?
I would not say reservation. It was more basically, because I always thought that if you do research here at the university here, this also should be something where you can, because this is basically the post-tax money or grants that we raised there, that would be something that you have to give back also and make something useful out of it. So that was always my intention of my research that I did at the university. And so we thought, no, we just have to try it now. And of course, if we had failed, then there would be always the opportunity to then go back to academia. So, but we thought, no, if we don't try it, then who will pick up those ideas that we develop? No one. So we thought, no, we have to really start and do it by our own. Mm-hmm. And that's how we did it. And that's great. We now have the first program in the clinics. It took us some time.
Sure.
But it's a complex thing, to develop pulmonary messenger RNA therapeutic. Yeah.
For sure. And we'll talk a little bit about that clinical program in a little bit. But from the business perspective, it almost sounds like a bad joke. So two scientists are in a bar, right? Doing mRNA research. When you decided that you were going to launch a company, you said you didn't really have reservation because you knew that you wanted to make a contribution with what you learned.
Yeah.
But building a company is a different thing from being a research academic or a professor for that matter. So what I guess maybe not reservations, but what were some of the early steps that you took around learning how to be at the helm of a biotech company?
And the first thing is to write a business plan, basically. That was the starting point. And this was basically, I have to say, that was also a trigger then, basically. We had in Germany is also business plan competitions, basically, for young startups. And then we thought, okay, we try to just put down to paper our thoughts, basically. And then you get, of course, structured thinking around, so what you all need and what kind of value proposition, what kind of drug, and to start to create a financial plan, a budget, and all these things. And there, I think we self-trained ourselves mostly, I would say. And then we thought, no, we will try it now, and we will do it. We will learn while we do it. Yeah. So that was the starting point.
Yeah. Did you have support resources? So like here in the States, I'm not sure what the biotech scene looks like in Munich, but here in the States, oftentimes startups will take advantage of communities, the incubators, even parent companies that spend biotechs on. So I'm just curious, like to what degree did you have the opportunity to lean into folks who had founded biotech companies?
Yeah, so there was definitely some exposure, because our first business angel who helped us also to move this forward and look into the right things that you have to basically consider. Yeah, so that was, and then there's, of course, also in Munich, we have a kind of small biotech hub there, with their kind of infrastructure. And this was also good where we got some exposure here to this creating a business. Yeah.
Yeah. Getting back to the science that you founded the company on, you said it started out with research in cystic fibrosis.
Right.
But by the time you realized that you had something that was worthy of building a biotech around, what was sort of your therapeutic intention at that time? Like in terms of indications and targets, did you think, well, we can move this well beyond where it started?
Yeah, it was always this focusing into the pulmonary space, because that's where I had my home, my scientific home, so to speak, and where I knew this field very good, the biology, and of course the exposure to thinking of what you have to consider for delivery, etc. So, and there are really a few severe diseases, pulmonary diseases, where there's not a good cure available. Yeah, so we thought, no, that's a good place to go. Basically, we did also some work in those days in bone regeneration, to locally. Translate proteins that are engrafted on collagen sponges to induce bone healing and non-critical defects, where bones are broken and they don't know anymore that they belong together. So you have the help there with biology. Then we did also some work in this space, basically, but mostly we then decided to look and go into the lung space. Yeah.
Mm-hmm. Okay. Remind me what time frame this was, when was Ethris founded?
So we started to look into using messenger RNA. That was roughly around the 2006, 2007 time frame. Yeah. And we followed the initial most important patents of our technology in 2009. Yeah. And then we started the company in towards 2011, basically, and really truly operational we became in 2012. It's pretty much 10 years from now.
Yeah, that's relative to the recent I guess advance in awareness and even growth, just in terms of the industry and the number of companies who are playing in this space. Those were early days.
Yeah, those were the very early days. And also, in the academic setting, basically, there was pretty, I mean, I think pretty much a lot of the birth of the messenger RNA drug modality, I think this came really from Germany. Yeah. So we had the longest there, I think that were our colleagues from CureVac, basically, Ingmar Hör, who were the first one who tried to develop messenger RNA for cancer vaccines. Then we had Ugur Sahin in the same time, the founder of BioNTech, who you were thinking of developing messenger RNA for the cancer treatment. And it was us, basically, I think, to develop our pulmonary therapeutics. So this all roughly in that time frame, basically. Yeah.
Yeah, that's fascinating. When in those early days, you mentioned your business angel, your angel investor. From there to sustain the company and to grow the company, what was the investment scene like in this therapeutic modality in those early days? Was it tough?
Yeah, that was tough, basically, because there was not this awareness of messenger RNA. Now everyone knows what messenger RNA is, of course. In those days, rather not. And there was also a time, where with the siRNA therapeutics, there were waves that goes up and down, And so there was sometimes some enthusiasm, and then I remember a time when Roche stepped out of their siRNA, then there was again less appetite, and so then it took a little bit again to create the awareness. Yeah, but this was early. It was really entirely new in those days. Yeah.
So how did you, as an academic and founding the company with an academic, beyond that angel investor, how did you learn to navigate the biotech fundraising scene? Like, what did you do in those early days to maintain, keep the lights on, right?
Yeah, speak with a lot of people, basically, yeah, and going to meetings, to conferences, and to find investors, and that's what we then also found, basically, our first investors who then are basically invest into the company so that we could really do the first steps. Yeah.
Yeah. Did you see a spike in interest from the investment community during COVID?
Yes, I think so. There was because of the awareness then, that we got from the mRNA space, and that I think it's very important for the field. I mean, in the end, the COVID pandemic, that was a nightmare for the world, but for mRNA, it's a great success, and that they could really, it could be shown that the full potential of messenger RNA, that you can develop really fast drug products, and most important, we have the approved mRNA vaccines. Yeah, so it's also now clear, that this is a new drug modality that can achieve approval. Yeah, so and I think that's very important and for the entire field now. Yeah, so and we have so many new great areas where people now explore the potential of messenger RNA as a new drug modality. So that's fantastic to see. Yeah.
Mm-hmm. Events like that. I mean, obviously, a global pandemic is going to have an impact on biotech, not just in terms of like the repercussions that we saw, right? Accessibility of raw materials and outsource capacity, that kind of thing. But it can also alter the course of the company's, I guess, pipeline ambitions. We saw during COVID.
Yeah.
This is hundreds of biotechs that didn't have COVID therapeutics suddenly did.
We had a little bit the same, of course, and we embarked on one program at the beginning of the pandemic. We're with a partner, a company in Switzerland, Neurimmune, which are incredibly knowledgeable in identifying human antibodies. Yeah, and so we teamed up basically, and they then from the first patients that were accessible to them, they isolated and identified and really super potent human patient-derived antibodies. Yeah, and the idea was that we translate this back, the protein code, into an mRNA code. yeah, which can then be inhaled, and you produce the antibody in the lung directly at the site of virus infection, so that you have, so to speak, a protective level, a level of layer of antibodies there covering the lung tissue. Yeah, and this worked very well. Yeah, so we could really nicely, you know, in those days, hamsters were used as a model, and we could nicely reduce and avoid this weight loss that you experience after infection with SARS-CoV-2. And we really moved this pretty far. But then what happened all in a sudden, and that's what we also know, this virus changes its face quite rapidly. Yeah? And then when we reached this Omicron level, basically, of mutation, we then unfortunately saw that the antibody was not neutralizing the virus anymore. So we unfortunately had to terminate that program. Yeah. So that's for one. And the second one is basically, you know, the program that is now moving about is in the clinics now. We just started the first phase 1 trial just before Christmas. This also a little bit goes back basically to the pandemic time, because, you know, that's a concept, ETH47, where we have a messenger RNA that encodes for a type 3 interferon, yeah, and these type 3 interferons, the lambda it's called, yeah, it is capable of programming the epithelium into a very broad antiviral state, yeah, which interferes with replication basically of any respiratory virus. That's a very broad activity, this molecule. And this basically goes also back to the pandemic time, because we got big funding for this program from the Bavarian state. Yeah, so who were looking in those days not only to put the bet on only vaccines, because no one knew in those days will the vaccines make it, but we also need therapeutics basically. Yeah. So in the end, the pandemic had an influence on what we did.
Yeah, it sounds like you had a win and a win and a loss, maybe. A bigger win than loss.
Yeah, absolutely. Bigger win, definitely. Yeah.
What was your pipeline structure before those two opportunities presented themselves?
Yeah, this was mostly centered only on our rare pulmonary diseases. Yeah, so that's still a focus of our company. You know, because for a program here, what I just mentioned, ETH47, this is a program where you need really a partner with a global footprint, yeah, to develop this drug, basically. But, you know, as a small biotech company, we really want to become a fully integrated biotech company that develops, manufactures, and sells its own drugs. And that, of course, is something you can achieve in a space where you have a sizable patient population that you can serve by yourself at the biotech company. And that's, of course, the rare disease space. So this was always our goal. And that's why our major focus is on rare genetic pulmonary diseases. So that's...
It's interesting. Well, you know, it seems the exception to the rule of late. Like most of the companies that I talk, most of the CEOs that I talk to are very clear about their intentions, and it's decidedly not to build their own manufacturing facilities and become an integrated biopharmaceutical company.
Okay, if you in the end fully manufacture everything in-house, that's a different question. You can use, of course, wonderful CDMOs. That's what we do right now. But we established all the manufacturing process there in-house. And that's also important, you know, because we have for pulmonary drugs, you have rather higher doses, you know, than for vaccines. So we always looked very early on in creating and developing our easily scalable manufacturing process. So and we have now a process developed that is not based on HPLC, yeah. So it's just built on tangential flow filtration, which is easily scalable, yeah. And it has the advantage that it's purely aqueous, yeah, no solvents involved, and that and this is what we transferred to the CDMO. We work with a fantastic CDMO there together who manufactures our drug product here.
Yeah. I'm interested in your perspective on the explosion of the anything RNA space. We'll stick with mRNA, but I mean, you can put any letter you want in front of it, right?
Yeah.
In a COVID and post-COVID world. And I mean, I think about it like you've been working on this science and this technology since, the mid 2000s and before. And for years and years, I'm not going to say it's going unnoticed, but it's not necessarily, in the media cycle. And then all of a sudden this pandemic happens. And then on the heels of the pandemic, it's like being the only person in the world who thinks the color blue is cool, or even knows about the color blue. And then all of a sudden overnight, like everyone's like, blue is awesome. We love blue, right? I mean, there had to be kind of you had to be aware of it, right? Like the explosion of interest and the explosion of companies. I mean, there's so many companies in this space right now. So what's your take on that? Like,
it's great for the field, basically. So it's fantastic. So the more approaches we see, the better we get. And there are so many diseases that can be potentially treated with messenger RNA. This can't be done by only one party, basically. And that's great that we have so many different approaches here, not only for the vaccines. So and I think we will see some new also potential approvals rather shortly, also for additional vaccines in the infectious disease space. But also look into the cancer space. I think we see really very nice good signals in this space where people use messenger RNA as a drug modality. But then look into genome editing. There's a great potential for messenger RNA to be used. And I mean, we have the first success now for CRISPR being approved, CRISPR technology. So that's a huge step, basically, and I think a huge stimulus also for moving this forward by using messenger RNA, basically. And then, of course, there's this field of rare genetic diseases where we see really, I think, pretty good progress. Also for liver genetic diseases, and of course we want to achieve the same with our pulmonary programs, and to really offer, hopefully as soon as possible, some help for the patients suffering from these diseases. So I think there's a lot really moving forward right now.
Yeah. In the context of your pulmonary focus, what do you see as the biggest potential risk to progress? What's whether it's a technical risk or a market risk or a scientific risk, like what's the rate limiting factor or big risk that could stand in the way of progress?
I mean, in the end, it's always the basic concept for developing a drug. And this all centers around right biology. You have to really understand your biology, then right exposure, so that you have the right PK. You know that your drug gets there where it needs to be for the right time, basically. Then, of course, critically, the right safety. That's all the basis of a successful drug in the end. And that's what you mentioned, is there the right patient. You have to understand your patient, basically, and then the right commercials. So I think it's a kind of umbrella, basically, that you have to look at and go into very detail for each of these elements, basically. And we did this, for PCD, for primary ciliary dyskinesia. We believe that we hit those boxes, basically. So we tackle very interesting biology, because in those patients who suffer from the disease, they miss certain proteins that drive the motility of the tiny hairs in our lungs, in our airways, that are responsible for moving the mucus out of the lung. So that's called mucociliary escalator, and it's made to remove any dust that we inhale from the lung, so that we have a fully particle-free surface of the lung. But what is important to understand here is that these cilia, they are just made by the airway cells once in the cell life, yeah, and the half life or the life of the airway epithelial cell something like 4 to 8 weeks, and that means you can imagine, yeah, if you deliver a messenger RNA during the ciliation process, yeah, you have quite persistent effect, yeah. So you translate a quite short-lived drug itself. The mRNA is gone in the lung after whatever, 2.5 days, something like this, into a quite persistent effect. Yeah, and so we believe that's a good starting point from the biology. And that's why we, for instance, picked the PCD. The patients is very clearly defined because it's a genetic mutation. So you take this risk out of it, basically, of the equation. We know that we need to potentially restore the ciliary function in about 20% of the ciliated cells. We believe that's doable from what we see in our preclinical studies, and there's a commercial potential because it's a life-limiting disease basically here, and it's a high unmet need because there are no drugs on the market. Yeah, so that's why we believe that's a good spot for messenger RNA therapeutic. Yeah.
That's, I mean, this is I could go down a rabbit hole here. Am I hearing you correctly? Like you talk about this ciliary regeneration, like
exactly.
cycle, and you're saying that that there's an efficacy opportunity at a specific point in that cycle.
No, you just have to restore the function in the cilia. Basically, and once the cilia are restored, you have a quite persistent effect, but because the cell does not change the cilia, yeah? So it's once built, and then they work until the cell is not is removed from the body and replaced by a new cell. Yeah, so for the lifetime of the cell. Yeah.
Gotcha. Okay. See, these are the questions I should have been asking in AP biology before I dropped the class my senior year. Not long ago, I had your senior director of formulation and aerosol research on a live event that I hosted for Bioprocess Online, Christian Doman. Great guy, by the way.
He is fantastic, I tell you. Yeah, absolutely.
Yeah, like he was super valuable. Just brought a whole lot of wealth and wealth of information to that conversation. But one of the things that we talked about quite a bit on that during that conversation was LNP stability and some of the challenges around there. So is that? I mean, do you see that as potentially a rate limiting factor?
I see that this limitation in the field, because I mean, just look into the commercialized vaccines, mRNA vaccines. There, so first we see that they need very cold temperatures to be stored, and that was okay during the pandemic, basically. Yeah, but now we move into the post-pandemic phase, and we really want to have our temperatures that the pharmaceutical industry is used to use, and also for the patient. Yeah, so that means this ultra cold chain is needs to be improved and need to be overcome. And I think we have done a lot here with our technology where we can contribute to this and achieve this. With our technology, we have, for instance, the lyophilization process in place, where we see that we can store the drug product already for a year. In the fridge, 2 to 8 degrees, and the studies are ongoing, but we don't see any change here. And even at room temperature, we see this already for more than half a year. Yeah, so that's I think a huge step forward. Yeah. And then also you have to see at and look into the stability of the drug product itself. Yeah, look the commercialized vaccines the ready-to-use drug product, so can be used for a few hours, basically. And we have with our snaps, what we call our LNPs, we have already achieved 6 days, at room temperature. That means that doctors can now really plans for using the drug for an entire week. Yeah, so I think that's a large improvement. Yeah, and on top, we have developed we call it stabilizing excipient technology. Yeah, those are a certain class of excipients from the inactive ingredient list, from the FDA that you can add, to the LNPs, and you make them mechanical stable. And why do I mention it? Because on the label of the commercialized vaccines, it's mentioned, don't check them, because they are very prone to aggregation, basically. Yeah. So, and we can really now stabilize these nanoparticles so that you can even vortex them, shake them full speed, and we don't see any changes in the particles. And of course, this gives confidence to also the doctors and pharmacists to use the drug, because they don't have any concerns anymore that might potentially aggregate. Yeah,
sure.
So, and also for manufacturing, when in particular the fill and finish step, where these nanoparticles flow through little tubings to basically go into the vial. Then of course you have to avoid aggregation to avoid any batch failures. So, and I think that's really important what we have achieved here. And then I think another, when we speak about limitations, I think another limitation, maybe, and I think that's a little bit unspoken also in the field, is that the formulations from the commercialized vaccines, they biodistribute to the entire body. Yeah, from their the documents from their authorization, you see that 30% goes into the liver. Yeah, it goes into the heart, even if you just locally inject into the muscle. Yeah, and even 2 to 4% of what's found in the plasma is found in the brain, basically. Yeah, and we think, if you don't need it in the brain, one should not deliver it to the brain, basically. And our snaps that are
seems logical. But...
Yeah. Okay. And our snaps that are made from cationic lipidoids, not cationic lipids, they are a little bit more positively charged, so they can package the messenger RNA more tightly. Yeah. So you need just half of the formulation, but they lead to a full retention of the mRNA drug at the site where you deliver it. And that's, of course, so you can avoid this biodistribution. And this is, for us, for our pulmonary drugs that we develop, we want to have the full activity of the messenger RNA, of course, at the site where it's needed. That means in the lung, and avoid any biodistribution. It's also a safety aspect, of course. Mm-hmm. So that's why we think really we have achieved pretty much here to move the field and our products also really forward with the advantages and to try to overcome those limitations that we just spoke about before.
Yeah. What's been your experience with I guess on the regulatory scene? Mm-hmm. So I have conversations with my colleague Anna Rose Welch, who's leading a new project at the company called Advancing RNA right now, and we often talk about some of the uncharted regulatory waters in this space. So what's been your perception there?
I mean, I think overall all the regulators are really very, very well educated now from the commercialized vaccines. I think that's a great step. Yeah. But what I think for the field is or would be very helpful, but that's ongoing already. Is to consider a kind of platform approach. Yeah. So where you can more easily, if you leave everything the same in your drug formulation, the mRNA formulation, but just then replace this with another messenger RNA, so that you get there certain
efficiencies.
Exactly. Sure. So this is for us also very important, this approach, because these primary ciliary dyskinesias, they are not caused with cystic fibrosis by only a mutation in one gene, but there are more than 50 genes known when mutated that can lead to primary ciliary dyskinesia, because you can imagine this is a very complex proteinaceous apparatus to get this beating ongoing. And for us, and it would be very helpful to just swap out one messenger RNA for another messenger RNA to then serve another genetic mutation that causes the disease. So that's those things that would be really helpful. But I think there's a lot of thinking into this direction already.
Yeah. When I look at like the ATMP space, cell and gene therapies, there's a lot of bad news. I mean, there's a lot of consternation about accessibility and cost, and even commercialization. I mean, there have been some not so smooth approvals in terms of commercial access. How does the mRNA field collectively ensure? I mean, obviously with the COVID vaccine, it was sort of a non-issue, right? We all got our shots. But how does the mRNA world, as it addresses more complex indications and perhaps charts some complex manufacturing waters, how do you make sure that you're not making some of those same, I don't even want to say mistakes, but trying to maintain an efficiency that results in patient accessibility at the end of the day?
I think you're referring to those gene therapies, where upfront you pay a few millions basically for a treatment. And I mean, the situation, I think, with messenger RNA drugs is a little bit different because it's more like a standard drug, that you repeatedly administer to the patient. So, I mean, for our PCD programs, basically, we believe that's a little bit into the direction that you see for cystic fibrosis, where you have already the pricing then here on the market.
So you don't anticipate any indications where that cost control might become a factor?
If you use messenger RNA, of course, for genome editing, that means a one-time treatment and then you are done. Basically, you face the same problems. But I think also the people start to think creatively how to solve this, what you mentioned. How can we find financial models, basically, that we can also serve those patients suffering from these rare diseases? Because in the end, for one rare disease, just a few patients. But if we sum up all patients that suffer from all the genetic the rare diseases that we have, those are many patients basically. And I agree with you, but I believe that the field is looking into creative reimbursement models to find, because we have to find a solution there, because we want to provide medicine to these patients as well. But for the mRNA, I think it's a little bit different because it's not a one-time treatment, or at least what we do, and then you fix it, but it's dosed like a standard drug, basically.
Yeah, very good. Just a couple more questions, then we'll need to wrap up. But I'd start with give us a clinical update, what the next big steps for Ethris are.
Yeah.
Like what's on your immediate horizon?
Yeah, exactly. So for us, important, and I mentioned this already, for the first program, we just started our first phase 1 study just before Christmas. So this is our study that has 3 arms, basically. One arm is where we deliver into the nose, and one arm is where we deliver into the lung. And then we will have, after defining their highest dose, basically a combined arm. And we do, of course, safety. That's their major clinical endpoint. But we look into also target engagement, which is very important, so that we can measure in nasal swabs and in sputum, from these treated healthy volunteers, the production of the recombinant protein produced by the messenger RNA, and more important, that we also see the downstream activation of the genes that are induced by the interferon lambda, which gives us the indication it works. It is really doing what it is expected to do. So, and this study we will have completed towards our springtime. So we think we will plan an interim readout towards early spring, basically. So that's the first very important news that we expect then in 2024. And then afterwards, we are also in the midst of putting the preclinical package together for filing a CTA towards autumn this year for our first PCD candidate. And so it's exciting time.
For sure.
At Ethris.
Yeah, very good. And then just to wrap things up, when you come to an environment like a giant investor conference at this stage in Ethris's sort of continuum on the clinical path, what's your MO here? What's your goal? What are you looking to make happen?
Raise money. No, seriously. I mean, now we enter into this clinical scene basically, and then we need many more financial resources than before. Yeah, that's our goal here at JP Morgan, and also to find partners. Because we have a really powerful, we believe, best in industry platform technology, and of course, this can also enable and help other potential partners to develop new drugs with our technology. So it would be great to leverage on top our technology also outside our focus area, basically.
Yeah. Excellent. Well, I wish you well the rest of your time here, and I appreciate you coming all the way from Munich solely for this opportunity to be on Business.
Of course, Matt. Yeah.
It's absolutely my pleasure. I enjoyed conversing with you and learning more about what Ethris is doing.
Same here, Matt.
Yeah.
So many thanks for having me.
Thank you.
All right. Very good.
I'm Matt Pillar, and you just listened to the Business of Biotech, the weekly podcast dedicated to the builders of biotech. We drop a new episode with a new exec every Monday morning, and I'd like you to join our community of subscribers at bioprocessonline.com, Apple Podcasts, Spotify, Google Play, or anywhere you get your podcasts. You can also subscribe to our never spammy, always insightful monthly newsletter at bioprocessonline.com/BOB. If you have feedback or topic and guest suggestions, hit me up on LinkedIn and let's chat. And as always, thanks for listening.